Welcome to Evolutionary Biology. Today, we are going to explore one of the most beautiful and unifying concepts in all of science, known as Neo-Darwinism. If you have ever wondered exactly how the physical traits of living creatures adapt and change over millions of years, you are in the right place. We are going to build this understanding from the ground up, step by step. Now that we know what topic we are tackling, let us understand what the modern synthesis actually is. Imagine Charles Darwin as a judge at a baking contest. He could see which cookies tasted best and were selected to be baked again, which is natural selection. But he had no idea how the recipe was passed down. Decades later, Gregor Mendel discovered those recipe cards, which we now call genes. Neo-Darwinism is the brilliant moment when scientists combined Darwin's survival of the fittest with Mendel's genetics. It teaches us that evolution is not just a vague idea, but a specific, measurable change in allele frequencies within a population over successive generations. Building on this beautiful synthesis, we must ask why understanding Neo-Darwinism is so absolutely critical today. It is the foundation of all modern biology. Without it, we would be defenseless against medical crises, like antibiotic-resistant bacteria. When doctors prescribe antibiotics, they apply a strong selective pressure. Neo-Darwinism explains exactly how surviving bacteria pass their resistant genes to the next generation. Furthermore, this framework secures our global food supply by helping us breed crops that withstand harsh droughts. It even gives conservationists the predictive power to manage small, endangered populations and prevent tragic extinctions on our constantly changing planet. To truly see how this evolution happens, we need a baseline, which brings us to the mathematics of population genetics. The Hardy-Weinberg principle gives us a hypothetical population that is not evolving at all. Our first formula is P plus Q equals 1. Conceptually, this represents the entire gene pool for a specific trait. The variable P represents the frequency of the dominant allele, while Q represents the frequency of the recessive allele. Because these are the only two versions of the gene in our simple model, their combined frequencies must add up to exactly 1, or 100%. If natural selection favors the dominant trait, P increases, and Q must decrease by the exact same amount. Now that we have the allele frequencies, we can expand this to look at the actual living individuals. This second formula is P squared plus 2PQ plus Q squared equals 1. It tells us the frequencies of the genotypes, which are the pairs of alleles an individual carries. The term P squared represents the homozygous dominant individuals, who inherited the dominant allele from both parents. The term 2PQ represents the heterozygous individuals, carrying one of each allele. Finally, Q squared represents the homozygous recessive individuals. Just like the alleles, the sum of all these genotype frequencies must equal 1, representing the entire population. When evolution occurs, these specific frequencies will shift. Before we introduce any evolutionary pressure, let us see this with a concrete example to establish our baseline. Imagine a population of 1,000 wild rabbits. Brown fur is dominant and white fur is recessive. We observe that 160 rabbits have white fur. Because white is recessive, these rabbits must have the homozygous recessive genotype. We need to find the frequencies of the dominant and recessive alleles. First, we divide the number of white rabbits by the total number of rabbits. This simple division tells us that exactly 16% of the population physically displays the recessive trait. Therefore, our value for Q squared is 0.16. Now that we have our Q squared value, we can derive the underlying genetic frequencies. This step is crucial because it transitions our focus from the physical rabbits back to the hidden genetic pool. By taking the square root of 0.16, we mathematically isolate the variable Q, giving us 0.4. This means there's a 40% chance a randomly selected allele is white. Next, we use our first simple equation. We subtract the recessive frequency from 1 to find the dominant allele frequency. The result is 0.6. We have now mathematically proven our baseline. The dominant brown allele makes up 60% of the gene pool. With our baseline established, let us introduce the core mechanism of Neo-Darwinism, a selective pressure. Suppose a new predator, a hawk, moves into the meadow. The white rabbits stand out brightly against the dark dirt, making them much easier to catch. Because they have a lower chance of surviving and reproducing, we assign them a reduced fitness value of 0.5. This means they are only half as successful at reproducing as the brown rabbits. The brown rabbits, with their perfect camouflage, retain a maximum fitness of 1.0. This environmental change sets the stage for evolution to occur. To measure the impact of the hawks, we need a new formula to find the average fitness of the entire population, denoted as W bar. Conceptually, this formula calculates how many individuals actually survive to reproduce. We take the initial frequency of each genotype and multiply it by its respective fitness value. The homozygous dominant and heterozygous brown rabbits are multiplied by 1.0 because they survive at maximum capacity. Conversely, the homozygous recessive white rabbits are multiplied by their reduced fitness of 0.5. If the environment becomes even harsher for the white rabbits, their fitness value would drop further, pulling the total population average down with it. Let us apply this formula to our rabbit population to see exactly what happens. We are given our initial genotype frequencies from our baseline calculations. P squared is 0.36, 2pq is 0.48, and q squared is 0.16. We multiply these by their fitness values. The brown rabbits contribute 0.36 and 0.48 to the surviving pool. The white rabbits, however, only contribute 0.08. Adding these together, we get a final average fitness of 0.92. This number is less than 1, physically meaning a significant portion of the white rabbits were permanently removed from the breeding pool. Now that we know how much of the population survived, we can calculate the new frequency of the recessive allele, denoted as Q new. This formula comes from carefully counting the recessive alleles that actually survived the hawk attacks. In the numerator, we take half of the alleles from the surviving heterozygotes, because only half of their genes are recessive. We add this to all of the alleles from the surviving white rabbits. Finally, we divide this entire sum by the new average fitness, W bar, which we just calculated. This division normalizes the frequency back to a standard scale of 1. If the hawks eat more white rabbits, the numerator shrinks and Q new decreases. Let us plug our numbers into this final equation to find the new allele frequency. We know the surviving heterozygotes contributed 0.48, and we take half of that, which is 0.24. The surviving white rabbits contributed 0.08. We add these together to get 0.32 in the numerator. We then divide this by our average fitness of 0.92. The math gives us a final answer of approximately 0.348. Physically, this means that after just one generation of intense hunting by the hawks, the recessive white allele now makes up only 34.8% of the total gene pool. So we've calculated our new frequency, but what does this actually prove? Let us compare our results. We started with an initial recessive allele frequency of 0.40. After the hawks applied selective pressure, the new frequency dropped to 0.348. The mathematical reasoning here is undeniable. The environmental pressure caused a measurable decrease in the frequency of the disadvantageous gene. This is the core intuition behind Neo-Darwinism. Evolution is not just a theory of survival. It is a precise, quantifiable shift in genetics. We have mathematically proven that the population has adapted in just a single generation. Now that we have proven the math, let us look at this diagram to visualize the entire Neo-Darwinian synthesis. On the top left, you see Darwinian natural selection in action, as a predator selectively eats the lighter, more visible prey. On the top right, we see Mendelian genetics, represented by a Punnett square, showing how those traits are coded by specific alleles. The magic happens at the bottom. The graph shows the frequency of phenotypes in the population. The x-axis represents the physical color, and the y-axis is the frequency. Notice how the original bell curve shifts entirely to the right over time. This visualizes exactly what we just calculated. The genetic makeup of the population has fundamentally moved toward the advantageous trait. To wrap up our lecture, let us review the most important ideas we have covered. First, Neo-Darwinism is the grand unifying theory that bridges the physical traits we see with the hidden genetic codes that build them. Second, we learned that evolution is not just a vague concept, but a precise, measurable change in allele frequencies over time. Third, by using the Hardy-Weinberg equations, we can establish a mathematical baseline to track exactly how species adapt to their environments. You have now seen how selective pressures mathematically drive adaptation. You have successfully covered the full mechanics of Neo-Darwinism, giving you the lens to understand the incredible diversity of life on Earth.